Now we'll move on to the next presentation. So late last year, there was an explosion on the internet about this one work that basically mentions that brain cells are really, really good in studying or not really studying, but kind of learning how to play the game Pong. And they did that using the Max 1 system. So I'm very happy that for today, it's kind of different from all the other speeches from this session. We have a speaker from Cortical Labs, Dr. Brett Kagan, who is the Chief Scientific Officer and has led the development of the DishBrain project, which we will talk about today. So this project established the first real-time synthetic biological intelligence system. Brett completed his PhD focusing on stem cell therapy before undertaking a postdoctoral fellowship in regenerative medicine and bioinformatics. Now, Brett's work is focusing on leveraging a variety of cross-disciplinary methods to investigate the fundamental basis for intelligence in biological neurons. So I would be very happy to introduce to you today Dr. Brett Kagan from Cortical Labs. Hello, Brett. Good
Hello
afternoon, good
there.
evening,
Thank
wherever
you
you
so much
are.
for that very kind introduction. And yes, it's a pleasure to be here and see all the great talks today.
Yes, so Brett is currently in Australia and streaming from Australia. So now I think we will be able to show your slides soon and you can start. Okay.
Excellent. Let's give this a go. Alrighty, thank you so much. It's a pleasure to be here. So today I'm going to be giving you guys a talk on structured real-time closed-loop stimulation and how this drives goal-directed activity in cortical neural cultures. So as mentioned, I'm Brett. I'm the CSO over at Cortical Labs. We're actually a small Melbourne-based startup that's investigating this idea of what we call synthetic biological intelligence. So if you're interested in this work, you can check us out. There's the website, and off we go. And so, what is the gist of what we're going to talk about today? I'm explicitly not going to focus too much on the sort of cellular characterizations that a lot of people done an excellent job at. Manuel, for example, excellent job on the sort of characterization of the cells. Then I sort of want to go sort of a step above that and talk about how might these systems work. And so we know that a living organism, we need what's called a closed loop feedback to operate. What does that mean? It means that when we do an action, if I reach over and I pick up a glass of water, I need to have feedback returned to me about the success or the outcome of my attempt to do that action, or else it's very hard for me to navigate the world. And they've actually shown this. So there's a great little study where they embedded rodents in a VR simulation, and they basically decoupled their action from their perception, and it led to these like stark disruptions in functional development. It's a great study if you want to go off and read it. There's a reference. And it's very obvious for us. It's almost hard to imagine a system where we may act in such a way where we do not receive that feedback as a result of our action. However, to implement it for an in vitro system is very challenging. And so that's what a lot of our work is. And so when we do achieve this, we call it an embodied neural network. Uh, and so, what does that involve? Essentially, it involves closed loop feedback, stimulating sensation. You measure the response in the culture of that sensation, then that does a thing, and you provide sensory feedback about that as you're going on. Sorry, one moment. I'm having a technical glitch. There we go. Sorry, something popped up on my computer that wouldn't go away. Where were we? Closed loop feedback systems. And there's been some great work in the past showing that these closed loop feedback systems can do stuff like increase plasticity in a culture when you use pattern multi-electrode arrays. But what's wasn't as well captured was actually a strong theoretical basis that may actually be underlying learning or the lack thereof in an in vitro system, if that was possible. And so I'm not going to go into a great deal of discussion about this because it is in itself an entire talk. But we essentially adopted principles from active inference via the free energy principle, which is something that's been championed by Carl Friston over at UCL, as well as a number of his collaborators and others throughout the world. And the idea of this, to state it formally, is that it's a process where you minimize variational free energy or entropy in a system through action and perception. What does that mean? It essentially says that a living organism, such as us, or even such as a collection of cells, needs to actively generate an internal model of the external world. So go back to that glass analogy. I, of course, as we all know who, with the neuroscience, psychology, so forth background, is we don't really act upon the real world, we act upon our perception of the real world. And so the goal then is to align that perception as closely as possible with the external world. And the way we do that is through feedback of how we receive it. And now, if I want to change and minimize the difference between these two, the real world and the model of the world that I have, there's two ways I can do that. One, I can generate a better internal model of the external world. Or two, I can act upon the external world, change my actions to make it accord with my internal model. Now, put simply, the way that we want to change and control our systems is to remove one of these two options. Okay, there we go. The clicker is limited. So basically, if we remove the ability to generate a better internal model by essentially turning the world into a random state, if a action is not selected as we would desire, we then limit a system to only be able to modify their own actions that would affect the external world to allow them to match their model. And so we essentially try to build an experiment out of this using the Max 1 MEA system. So we there were three different types of cell cultures we used. Predominantly, we used either human iPSC cells that we differentiated using the dual-SMAD inhibition protocol, which gives us nice mixed cultures. I think there's been several people who have shown very similar cultures throughout this talk like that. Or we use a directed differentiation process like the NGN2, or we took sort of more classical primary cell culture methods. We plated them on our Max 1 MEAs, and then we subjected them to a simulated game environment, which I'll discuss a bit more. The key thing to focus here is the actual feedback loop that we created. So there was input that we gave them, and then that input would tell them where the ball was. Under our regular, what we call the stim condition, which is most of the data that I'm going to be showing you today, if they missed the ball, we gave them unpredictable random sensory feedback. That is essentially information that cannot be predicted. If they were successful at hitting the ball, the game continued. Not only is then the direction of the ball predictable, but we gave them an extra bit of predictable sensory feedback. And now, under the theory that we're proposing, if cells are really trying to, or neurons or a neural system, however you want to phrase it, is trying to minimize its entropy or minimize the surprise, or even just model the world, however you want to phrase it again, the idea is they should avoid a state that is inherently unpredictable and gravitate towards doing things that lead to greater predictability. So perhaps it's a big ask for a couple of cells sitting on a dish, but we gave it a go. And then we wanted to follow up with a bunch of other control conditions. So we had our, what we call the silent, or SIL, represented as silent condition, which is where they were given information where the ball was. When they missed, however, we removed all of the stimulation. And the two analogies between these is the stim condition. You can think of it as they missed the ball, we gave them a painful electric shock. Obviously, there's no pain going on there because they're just neurons. There's no pain receptors, but for an analogy. For the silent condition, they missed the ball and we turned off all the lights and sound for the world. Then we had a no feedback condition, which is actually an open loop condition, just to see that is it actually the closed loop feedback aspect that's driving the learning, or is it simply they're going to try and hit the ball anyway because, you know, some sort of basic Hebbian plasticity and the correspondence of where we fire, who knows, lots of reasons. Finally, the last sort of sophisticated control we had was a rest condition where the cells were controlling the paddle, but no information was presented whatsoever. So this was the setup of our experiment. And the way that we ran it, here's just a very quick schematic. If you want, there's more information in the preprint. I believe the link for that's been shared, and hopefully the not real paper, but the peer-reviewed paper will be coming out very shortly, hopefully. Review processes, I think we can all know how that goes. And so essentially what we have here are the software components for this, what we call the DishBrain system. And so it interacts with real time with the Max 1 hub and the Max 1 MEA system, as well as the MXW server. And then we basically shuffle around selections of spike detections, environment that we built, as well as a stimulation and a LAN interface to tie it all together. And that plays out. And so this is an example, and great, the video starting automatically, of basically visualization of what we did. And so this is actually, you can go and look at a longer version of this visualization if you wanted, the link below. Hopefully this is coming through clear for you guys. Each one of these, as you'd be familiar with, represents one of the max 1 electrodes. You can see we've chosen a configuration here where the top longer rectangle is what we deemed the sensory region, and that's where we're putting in 8 different points of sensory information, which is topographically mapped to the ball relative to the paddle position. So formally said, it would be an egocentric representation of the game world. And then the 2 discrete regions at the bottom are the motor paddle. So as activity in those areas change, that then modulates the change of the paddle. And over time, as I'll show you a few results in a second, they get better at playing the game, which was nice to see. So here's a bit more information on how we actually set up the system. So we applied stimulation into those 8 stimulation points that are sitting at the top in the schematic. And then we had these, we tried a bunch of different motor regions, and for the most part, they were all the same, but we ended up settling on this sort of split configuration. When I say all the same, I mean, seems the neurons were very adaptable for however we wanted to lay the ground at. So this is what we picked because we had to pick something. And so we had ended up with these split configurations where essentially we measured and looked at the relative activity across the 2. And I think we've seen quite a few raster plots, but this is what it looks like when you're throwing sort of very low latency stimulation on it. This is actually just mostly on a control chip, so there's not too much background noise, so you can see what's going on. So here you can see the ball's been delivered. This is actually as it goes up the first time it hits it, you get its predictable stimulus. It goes down and then it misses, and so on and so forth. And that's how we sort of ran the game, and so then we tested it. Now, I'm not going to go, as I said, too much into the cell validation because there's been a lot of excellent work. There's more of this in the paper, and these methods we've used are all very well established with many, many papers sort of showing that they lead you to nice cultures. But just to set everyone's minds at rest, we did do this. The cultures, they displayed markers for neural cultures, morphologically neural cultures, neuron, beta-3 tubulin, MAP2 for neurons, axons and dendrites, respectively. And not only were they neurons, but they were at least a relatively high proportion of cortical neurons, along with supporting cells for our cultures. And particularly, we do Ki67 for when we're taking them from human-induced pluripotent cells, because we want to make sure we don't have any runaway aberrant differentiation going on. And here's a lovely picture of them using scanning electron microscopy integrated onto the Max 1 chip. So you get this deep long-term integration. This is about a 6-month-old culture, so still living, living before we fixed it, of course, and quite happily integrated onto the Max 1 system, which was brilliant. And then we did a lot of stuff that people have investigated, such as we made sure that these cultures develop nice activity consistent to what people have reported previously, and tracked that through a number of different cell cultures and made sure we had healthy spread across it. We used a relatively high density plating, so we tried to cover as much of the region as possible to make sure that there was connectivity of the dish as a whole. And then we investigated learning. So just to go over the groups we did for our initial experiment, we had they're down here. We had a control group, which control simply is media alone, so a Max 1 chip sitting there with media only. We had an in silico group, which was actually without the chip in this case, so it was a computational model, just with the paddle being moved by random white noise on a Gaussian distribution. Then we had our rest condition here, where the cells were actually controlling the paddle. They just weren't receiving any stimulation. They didn't know what was happening. It was just spontaneous activity moving a paddle. And then we had mouse cortical cells and human cortical cells, where we both stimulated them and reported from them. And I got to remember to use the clicker to move it. You can see here, if you compare it, these are a little hard to make out, these graphs, when you're not used to looking at them. But if you just compare, let's say, the top right corner of these graphs, you can see quite neatly that there's a far higher percentage chance of longer rallies occurring later on than at the start. And if you want to see that even more clearly, you can just break them up. Pilot data sort of showed the learning curve for these cultures, at least, seemed quite prevalent around 5 minutes. And so we just set this as a sort of a priori test that we're going to do. Look at the first 5 minutes, look at the last 15, see if there was any learning occurring. And we saw that for the control and the in silico groups, as we would hope for something with no cells and merely white noise, there was no learning. For the rest group, we saw again no significant degree of learning, which is what we'd hope since the cells were not seeing anything. However, for both mouse and human cortical cells, when they were learning, we presented the information. Sorry, what I should say is when they were presented the information, they both showed significantly higher learning over time. That was also then significantly higher than all of the controls. And that was really exciting. And there's a lot more of this data if you want to go look in the paper. So what I want to jump now to is actually the fact that not only did we see this learning, but it mapped to really interesting activity changes. And so what's unsurprising for, what should be unsurprising for everybody, is that whether you're looking at the rest during rest, so no activity being applied to these cultures, or gameplay when activity is being applied to these cultures, there was a significant correlation between the two motor regions and the sensory region, which is what you'd expect for a culture. What is interesting and particularly surprising if you look at this first graph is that actually when they're playing the game, the degree at which that correlation is, so if you take the average of all of the cross correlations, then you see that there is significantly more correlations or coordination, I should say, between the regions during gameplay than when they're at rest. And this is, of course, exactly what you'd expect if there was some sort of overall regulation of activity going on in the dish. And you can see this starkly in another way, where if you look at not just the coordination, but let's say we're going to look at what we call the exclusive motor region activity, where you're going to get activity in, let's say, the left region, but not the right region at the same time. Now, this would also have to occur and be increased if these cells were actively controlling the paddle and in either inhibiting one area or increasing another area to fire, to actually have an active control. You'd have to see more exclusive activity. And we see that between rest and gameplay, there is a stark increase. And actually that this correlation between them decreases significantly in about the first 5 minutes, which is exactly what we sort of were seeing, where the sort of learning was the biggest part of the learning curve was happening. And then it stabilizes and it stays at a relatively low correlation, which is, again, all really nice and supportive and provides sort of a mechanism of what's going on in the dish. But to take it even a step further, which has totally fascinated us, and there's no preprint of this out at the moment, and there's a lot more data around this, but it will be going, it is very close to being completed and finalized, this data, is this investigation of what's called criticality. And so for those of you who aren't as familiar with criticality, it's essentially a state where the population of neurons is at the borderline between, say, strong and weak coordination. So you can imagine a system in a brain where cells are firing in an oscillatory behavior. It's very controlled and coordinated. Or you can imagine the far end where cells are firing in a completely random, uncoordinated manner. Well, the theory of criticality is that the brains themselves are wired in many cases and optimized to sit at criticality, especially when engaged in sort of cognitively demanding tasks. And there's been a lot of really great recent work, predominantly in vivo, finding that actually criticality is linked to responsive drugs. And even, my favorite is actually the scores of fluid intelligence that they found very recently were linked with the higher degrees of criticality. Now, it is a controversial area that's being investigated, but it's certainly interesting, and so it prompted us to want to investigate this. And what we found was some very stark results. And so here's just a quick, I'm just wary of time, so I won't go into too much, but the gist of it is when the cells are engaged at their rest behavior, they're showing metrics that are further from criticality than when they're actively engaged in the game. Next slide, please. There we go. And this is incredibly stark. So I don't have time to go into any more detail than this right now. But what I'll say is that we looked at a number of different measures of criticality, 3 key ones, which is sort of gold standard criticality analysis for anyone familiar in the area. And when we wanted to predict that, if we included the actual performance of the cells as a metric, we're able to predict whether they were playing a game or resting with a 98% success rate. Which is cool. Like, that's pretty interesting. It sort of says very robustly that there is like very different stuff going on in these cultures when they're playing a game or when they're resting. But more than that, and I think this is really exciting, even if we remove performance as a metric. Criticality alone using random forest was able to predict 92% success of which category these cells were sitting in, suggesting that really what we have going on here and how the cells are reorganizing their activity is not merely some is there sort of heavy in plasticity and stuff going on? Of course, very likely. But it's not merely something as simple as cells fire together, cells fire together, cells wire together, or you're just upregulating one area of activity in some sort of really gross and boring manner. Not that I think even if that was happening, it's particularly boring, but it suggests that these cultures are engaging in some fairly complex changes, which is truly fascinating. So the last key bit of data I'm going to show you guys is also what I mentioned before, is that we wanted to investigate that what we saw happening in terms of their learning was actually because of the closed loop feedback and the sort of stimulation feedback patterns that we give them underneath the sort of neurocomputational theories that I discussed before. And so you can see here, when you take your stimulus condition, the cells still continue to learn. We're able to replicate our previous result. However, when you remove the random feedback, their learning was greatly ameliorated. Interestingly enough, the silent group, despite not showing significant learning over time, did end up outperforming our no feedback group alone. And although that might seem counterintuitive for a second, one, it must be recognized that silent feedback is called silent feedback because it is actually feedback. Turning the lights off every time you make a mistake might not prompt you to learn and change your behavior as quickly as an electric shock, for example, but it might still be enough information for you to want to change some behavior. If you consider that learning meaningful or not, that's up to you. But there's also even the idea that when the game resets, the direction of the ball and therefore the stimulus pattern is in itself unpredictable. So that might also tie into the theory. But overall, I think this is very neatly shows sort of evidence of sort of a validation of our approach, that not only is it the feedback that's important, but the specific sort of free energy minimization approach certainly seems to be one way to drive learning and behavior. And so to conclude and sort of wrap up as a summary, we can sort of conclude by saying it's really great to see this DishBrain system does exhibit a sort of natural intelligence, but with an almost, we'd say, a self-organizing adaptive computation occurring within neurons. We were able to successfully integrate neural networks with the Max1 HDMEA. And then through this electrophysiological recordings, they were able to be embedded or embodied in a synthetic world, which stimulated this game of Pong. And this is actually in the right side. These are real neurons controlling the paddle. Of course, we admit it is a cherry-picked little rally length, but it's not an aberrant or totally bizarre rally length. We do see some beautiful rallies that occur. And yeah, I think we're very proud to say that we believe it's the first real-time method to harness what we're calling synthetic biological intelligence, and we think it's an incredibly adaptable approach to a lot of other things. We are continuing to work and develop on it, but we're excited to see where this technology goes next. And so I think I'll just end with this quote that I love, because I think it sums up a lot of what's going on. Because I know I've dumped a huge amount of data on people, and it's to say that if the human brain was so simple that we could understand it, we as people would be so simple that we couldn't. And I think that's that's beautiful when you consider the work that we're doing here, because what we're hoping to do is be able to start to simplify some of these incredibly complicated processes that occur when it's scaled up into a human brain. But perhaps by breaking it down and pulling it apart, we're going to be able to start to understand a little bit about what makes us tick. And maybe we can even start to use it in various ways. And so if you're interested in that, please feel free to reach out for collaborations. As always, this work would not be possible if not for the amazing team that we have. And also as a shout out to Maxwell Biosystems team, because with their help with us, they went above and beyond in their assistance with us, getting things set up, accessing their systems so we could use them in this real-time, low latency manner. Their help and support is always there, and it's always appreciated. So big thank you to them. And yeah, I'd be really happy to take any questions if anyone has any questions.
Really, really fantastic talk, Brett. Thank you so much. And yeah, same. We at Master Advice Systems are also super, super happy to be in collaboration with you. And I hope we can just move on and have some more activities together moving forward. Okay,
Yeah.
so now we open everything. We open the room for questions. We have one on site
Thanks
from.
a lot for
Mhm.
the talk. I also read the preprint.
Yes.
So one question is that the danger is that your sensory encoding directly influences your motor areas. So what would convince me that it's actually working if you could flip the arrows, the ones that drive the pedal up and down, after the 20 minutes training, and it would again learn the new pedal directions, and it would still be okay. Did you do that control? So you're flipping the pedal directions and then teaching the network with the same sensory encoding.
Yeah, we did, and in fact, in the supplementaries, it'll show you there's I think six different configurations that we used, and we saw similar performance from sort of four of them. We actually initially started with a very rough sort of left, just two regions. Left meant left, right meant right, and that was our worry with bias. But the other thing that we do that helps control that to an even greater extent is we actually do make sure that whenever we do deliver stimulation, we also blind our system, our dish server system, to the own stimulation. So we make sure that it's not being driven through sort of stimulation or bias alone, because that was one of our chief concerns to address. So yes, we've done that as a control. We didn't present it any more than in just the uh, in the supplementaries. As I said, there is one little figure there that sort of shows the different aspects in the supplementary data. But we have done it. But then more than that, like the important thing was actually building in good blinding systems, because they are very sensitive. And certainly when we started out, we saw amazing. We thought, oh wow, look, they're learning so well. And then we stuck on some media controls and went, oh no, look, they're learning so well also, and realized, nah, we have to fix this. And so yeah, it was something we addressed very, very early on.
Thank you. We have other questions from our online audience. The first one is from Joanna Kaksonoska-Gotz from Ahead Bio. So the question is, how is the sensory area connected to the motor area on the chip? I think when you showed these compartments on the chip, she's asking how are the sensory areas connected to the motor areas?
Yeah, sure. So the actual entire surface of the chip is covered with cells. So the only thing connecting them are the cells. Um, so that's essentially how they are. So we make sure, and we found, and we've explored this a bit more in the paper that's currently under peer review. But essentially, yeah, if there's not sufficient connectivity over the whole chip, then there's not a lot of learning, as you would expect. So that is a requirement, that you do have sort of fairly uniform, fairly high connectivity across a chip in terms of cells. But if you do, then they're able to sort of communicate with each other via the cells, which is what we were hoping to see.
Okay, so basically it's just a random network of neurons on the chip, and then you just assigned parts of it for the sensory and parts of it for the motor part. Yeah.
Absolutely, absolutely.
Yep.
And it was brought up to us that that was potentially a weakness, that we weren't looking at the actual electrode, electrical activity of the cells, and then purposely mapping regions to where the cells actually were. And that's how we started off doing it. And then we thought, you know what? The question we're really asking is, how adaptable are these cells? Uh, if we give large enough motor regions, and there should be some cells there, the question is, how much can they reorganize their activity? And so the motor regions, although not completely arbitrary, are are to some degree arbitrary. And so I think that's makes the results really interesting, to show that even if you are kind of forcing cells into boxes, they may not necessarily fit. I talk about here boxes in terms of the motor regions. As long as there's enough distribution across the network, they will reorganize their activity quite remarkably. And of course, that makes sense when you consider the degree that a in vivo, like an organism like us, will be able to reorganize our activity, regarding what sort of sensory information we receive. But it was quite remarkable seeing that in the lab as well, and it's what we hope to sort of show. And I think the results bore out that it was an adaptable system.
All right, thank you so much. Brett, we have more questions for you, but I think we don't have time now, so we're just going to move it forward to the roundtable time. So we'll see you
Absolutely.
later. Thank you so much.
Thank you.